You need to point in a random direction and hope that it gets you there. If you were a tinkerer, you'll probably do quite well in learning how to do molecular biology. Biology is all about thinking. It's what you have a puzzle and you're trying to figure it out, and you have totally incomplete information.
Hello and welcome to another episode of the Professor Mahesh Podcast, your resource to help you make informed career decisions. As we like to think, this is your future simplified. This podcast is brought to you by IITM Global, an initiative of the Office of Global Engagement at IIT Madras. Please learn more about their activities in the links below. IITM Global is an organization that is there to take everything IIT Madras and everything India to the global audience. Our teaching, our research, our startup zeal, our entrepreneurship, and our philosophy fundamentally. In today's conversation, we're going to dive deep into the world of life sciences. We all lived through 2 years of COVID. Everybody now understands the value of a vaccination if you didn't understand that before 2020. All of the vaccines, all of the work we saw was completely due to well-trained, passionate life scientists. This army of people saved millions of lives globally. If you want to join an army like this and still have a reasonably rewarding and satisfying career, where would you start? What would you do? That's going to be the topic of our conversation with Professor Mukund Thattai from the National Center for Biological Studies in Bangalore. Mukund, thank you very much for making the trek up from Bangalore on a rainy day like this.
Yeah, my pleasure to be here.
And let's start with your own sort of training, where you stand in the broad spectrum of biologists. You've made an effort to bring math into biology. That's been your passion. Tell us a little bit about your journey from physics to biology.
Yeah, so since I've been called to do the session on life sciences, I should start with the caveat that I was trained in physics. My undergraduate degree is in physics. My PhD is also in physics. And that's the reason why I tend to speak in the language of math when I engage with biology. But just to say that the intersection between math and quantitative reasoning and biology is very, very old. If there's an impression that biology doesn't even require mathematical language or formalisms to function, right? But if you think about for example, the huge challenge in biology in the 17 and 1800s was the challenge of classifying all living things. And if I told you or asked you which is easier, to classify all living things or to classify all chemical elements, clearly the former is much more complicated, right? The classification of chemical elements eventually ended up being this sort of two-dimensional periodic table, which itself is a huge achievement. But living organisms are much, much more diverse. They have many more traits to keep track of than just mass and charge, for example. And yet, the taxonomists and the naturalists of those centuries managed to put together a huge amount of measured data. And then they use these data to come up with a system of classification, which is this lineal classification of all living things, which is a tremendous achievement. And it's on the basis of that classification that Darwin had his first insight that all living things on Earth are related in a tree-like manner, which is also a profoundly mathematical statement.
Yes.
Yeah? So the idea of careful measurement and careful analysis of those quantities is central and has always been central to biology. The same goes for example, the revolution in genetics, which happened through the 20th century, was based on Mendel's studies of the inheritance of simple traits, which also could be summarized in simple mathematical language, right? And in the 20th century, the development of molecular biology, physicists played a huge role in this process. So you have, I think, multiple collisions between people who are not trained in biology but who are passionate about it, and then people who bring the data, and that keeps on happening. And we're living through such a collision even today. When I got started, I didn't know any biology. The last time I studied biology was in school in the 10th standard. And I did my undergraduate degree in physics, and at that time I was interested in things that, a physics student would be interested in. General relativity or condensed matter theory. Those are the kinds of subjects I thought I would be working on. And that's what I was thinking about when I got to graduate school to start my PhD. I joined thinking that I would do a degree in condensed matter physics or pursue research as a PhD student in condensed matter physics. And this was at MIT, where luckily there was, in my first year there, my second semester there was a reading course that was offered by a person in the brain and cognitive sciences department. His name was Sebastian Sung, who happened to be a physicist who ended up studying brains. And I thought this was interesting. And he was giving a reading course on cells and how cells can do computations. And so it was a physicist speaking the language of physicists, but telling you about how living systems work. So I sat in on this reading seminar. And that was really my first introduction to proper biology, right after 10th standard. And I was hooked at that point. So it was at that point that I decided to even think about doing a PhD on a subject which was at the interface between physics and biology. I was lucky enough that the MIT physics department hired their first faculty member who was doing work on bacterial gene expression. Right, so this is something that's hardcore biology, but this is Alexander van Oudenaarden was hired in the physics department, and I, along with two other people, became his first PhD students. And we built a lab. We built a wet lab in the physics building, building 13, and we started doing measurements and making mistakes. Obviously, we didn't know how to do cloning, and we didn't know how to splice genes or purify plasmids, and we quickly learned to do all those things. And so I was lucky to see the building of a lab very early in my PhD career, a lab where we were doing experiments that biologists would consider proper experiments, but we were also analyzing it using theory and models from physics. So that was my journey. And after my PhD at MIT, I moved to NCBS in 2004. So over 20 years ago.
You use the word proper experiments. Let's come to these proper experiments and also tell me what are improper experiments.
Biology has this sort of undeserved reputation for imprecision among people who don't know. If you are running a biology lab, you need the place to be clean, there's no contamination. All your reagents need to be at a certain level of purity and within tolerances. So this is something where if you want to do basic molecular biology, having all this working in the background really, really helps. Okay. So secondly, a biology experiment, if you go see a biology lab, and when students come to visit NCBS, they say, I want to see a lab. It's a bit underwhelming because you go to any biology lab and what you see are, tubes, and all the tubes contain clear liquids, and people are sort of moving clear liquids from one tube to the other. That's what it looks like, right?
Right.
You don't see a particle accelerator or a telescope or even a microscope in a molecular biology lab.
Right.
And imagine the discipline it requires and the planning and the logical precision to design the experiment. So you know that from this tube, you're moving to that tube and that tube. Each of these is there for a purpose. Even something as simple as extracting DNA from cells. You have to prepare the cells, you have to lyse their membranes, you have to get out the DNA, you have to separate the proteinaceous matter from the DNA. These are all very precise chemical steps. So it requires, of course, training, and it requires also the ability to troubleshoot experiments. These are things which you will not be able to get away with cutting corners here. Yeah? And you learn this very quickly because the first few times you do an experiment coming into biology lab, your colleague who's, let's say, trained in molecular biology, she makes it work immediately. And you do the same thing and it doesn't work at all. and it looked like you did the same thing. And you say, what did you do differently? And the answer is you're paying attention and you understand what's going on. I think this may be a good point in the discussion to say that if you're trained in the life sciences, one of the most important things for you to do is to go and do experiments, or at least go and make observations, which could be out in the field. Go and pick up something from outside and observe it and record your observations, at least that. So lab work, field work, observational skills, the skills to record and document, these are all things which are absolutely essential to doing biology. And you don't get this from the textbooks or a dry lab or a classroom.
And in this whole process, there are a set of skills that are almost necessary. You talked of, splicing, cloning, these skills, there's a perception that they take time, that one needs to, quote unquote, do your time before you start doing it. Is that close to the truth, or how does this whole system of training work?
No, I think unless you actually do it and make mistakes on the way, you will not figure out how to do even very simple protocols properly, because there's a lot of unwritten steps in these processes. You don't just get it from picking up a lab manual. You need to see somebody doing it, and then you need to do it also. There is a level of improvisation. You could, for example, say that somebody who's very good at baking, right? What they're very good at is taking materials that are not always very well defined. The flour will be different every day, and somehow getting the output to be the same every time. So it requires a little bit of tweaking on the way. You adjust the oven temperature, you adjust the batter mixture till you feel it's just right. These things you can't write down, but you need to go ahead and do it. I think the question you're asking, though, is, is it better to just have students very early on go into the lab and make mistakes? And I think, yes, it is.
Okay.
Because then they learn and they get better at it. It is not possible, in my opinion, to teach people that here's step 1, step 2, step 3, step 4. You see it once, or in some rare cases, you get to do it once, and then it's assumed that you know how to, let's say, do PCR. You don't.
Yeah.
So it really is extremely valuable to go do those experiments. There's the notion that biology experiments are expensive to do, and that's why people are not trained to do it. That is only true if you're using these commercially available kits, which are very expensive. Right. Reagents are very expensive. But actually, you can build very frugal teaching laboratories where you build up your own reagents and even your own machinery. You can even build a PCR machine for very cheap. You just need a few, a fridge and a microwave. Again, it looks like a kitchen. But as long as you get all the equipment and you get students to go and start doing these things and thinking about how they're making these buffers and solutions, what order they're doing things in, you can do this training at reasonable cost. And the benefit is immense, that people actually see why these buffers have these pieces in them instead of buying a kit.
Sure. You are making an important case here, a broader case of experiments in high school.
Yeah.
Be it physics with my old simple pendulum experiment or life sciences, I don't think that has changed in decades. Maybe with these low-cost PCR, we may be opening the door for fundamental revision of the high school science lab curriculum.
So I don't know the current state of the high school curriculum. I certainly know when I went there, when I was in high school and doing biology, we didn't do any of this. One thing I should say, though, is if you have a biology lab, whoever's running that lab has also additional responsibilities, because there are safety, biosafety issues, and keeping living samples and the disposal of biosamples and so on. So these are things which a school lab typically will need to work a little harder to do. If you're working not with living samples, but somehow you are able to still do sort of observations, you can do microscopy, of course. Right. That's something you can easily do in school.
Sure.
So I think it's eminently possible to create a very exciting high school level biology curriculum with experimentation and observation. And if it's not there, it should be done.
There's room for an entrepreneur to come think through this and maybe
I think there are people who are doing it. There are people who are thinking about what a good frugal high school teaching lab in biology will be.
Yeah, wonderful. Let's switch topics. In this day and age, in 2025, why should a bright young student take to life sciences
Oh.
when there are so many other persuasions, starting with, computer science and
Sure.
everything else?
Sure.
What's in it for a future life scientist?
So a student who wants to become a scientist or who already thinks they may want to
Or want to sort of go in this direction, correct?
So for me, the question is a no-brainer. But let me try and convince the skeptic. So I think if you look at the breadth of life sciences, there'll be something in there for everybody, which is, I think there's no other field that is comparable in terms of the diversity of approaches, the diversity of systems you're studying, the diversity of really big questions. I mean, the really big questions are, how did life arise on Earth? How do cells replicate? How does the brain work? How do social systems develop? I mean, these are giant questions that anybody will be persuaded are important. Of course, physics also has giant questions, right? So, but in the life sciences, we are still at a stage where somebody just getting into the field has a shot at jumping into the middle of a really fundamental question and making a difference. In physics today, such success requires a long period of training because you need to get to the state of the art, right? And in the life sciences, you can just jump in and start doing really important work in some new field that you just learned about 2 years ago, right? So you will be able to find something that matches your interests. In terms of course, a student thinking about a career, I think there'll be other questions you'll ask about later. For me, the reason I really love life sciences is because you get inspired about the work you're doing on a daily basis just by going around and looking at things that are happening in the world. It doesn't require a particle accelerator to generate the data that will get us to think about things, right? Even very simple observations about, why do certain species cluster together in some forest? You'll find very quickly that these are totally unexplained, and but the pattern is very robust. So you can start going and thinking about it. The field that I work on the evolution of complex cells, and this is something which I got to by accident, and maybe we'll talk about that later. But I think it's a very fulfilling type of field to be in and make a contribution, because if you answer a question about the evolution of life on Earth a long time ago, you can somehow see that it has implications for explaining who we are and why we are here and how we relate to the rest of the planet, right?
Sure.
So it's something which you're also able to explain to other people. Here's what I work on. Here's why I think it's exciting. It doesn't require a huge amount of domain jargon to explain what I do. Of course, you use a lot of specialized techniques to address those questions, but you can also deal with them at many different levels.
It's easy to explain to that old aunt or uncle,
for example,
as to what you're really going after.
For example, it's certainly easier than certain types of math or physics.
Sure. The allied question to that is, why should I choose a career in life science research or life sciences? We'll come to non-research careers as well, versus practicing medicine as a doctor.
So we should deal with one question right now, that human health and life sciences are related but totally different aspects of study and practice. People are under the impression that if you do biology, you're on the track to doing medicine. And that's wrong in both directions. You can actually become a doctor without an undergraduate degree in life sciences or an allied discipline.
Sure.
You need a medical degree. And conversely, 99.99999% of life sciences is not to do with human health. The example I keep giving when we do our student orientation at NCBS, whenever we have the incoming PhD batch, we talk to them and say, why did you get into biology? We ask them during the interview, and we ask them again when they arrive, right? And in many cases, people are interested in how the brain works, or how do ecosystems function, or I love field work. That also happens. But you do get a reasonable number of people saying, I had a family member who suffered from this disease, and I would like to do work that would contribute to the elimination of this disease. And their passion comes from that very personal story. But my response to them is, if you want to make any contribution to solving this disease, then you can't just point at that as a target and assume that you're going to be able to solve that problem. You need to point in a random direction and hope that it gets you there, right? Because these breakthroughs in life sciences come because of people who are studying very crazy esoteric systems for their own sake. Very famously, CRISPR, which now people use to modify genes inside our cells, came from people who were studying how bacteria have a rudimentary immune system, how bacteria evade their own pathogens, right? And the latest fad, which are these GLP inhibitors, this this weight loss, Ozempic drug, comes from the study of a toxin in a reptile, the Gila monster, right? Which nobody would start studying that with the view of developing a weight loss drug, right? So even if you're interested in having an impact in human health and translation, in an aggregate basis, the best bet is to have a large number of people doing a lot of different things. Then a smaller number of people taking examples from that which may have benefit for some sort of human health or disease, and then a smaller number of people developing that for commercialization. And so you need that pyramid, right? People think they can build a tower. Everybody's working on the disease. You need a pyramid where the base is working on everything. And so if you want to, even if your intent is to contribute to improvements in human health or planetary health or agriculture, right, all these very important topics, I would still urge students not to specialize very early onto those subjects. Certainly not during your BSc or BTech, and potentially not even during your PhD, but only later.
Okay. It's important, you argue, that people should have a broader understanding of the discipline. Now is a great point in the conversation to start moving towards taxonomy of life sciences itself. Okay. This, as you said, life sciences is a very broad discipline. Let's slice and dice it for the benefit of our audience.
Okay. It's a tough question, but let's give it a shot. So there are ways to classify it at, for example, the length scale of what you're studying, right?
Okay.
So let's try that, right? So there are people who study the molecules that make up all living things.
Okay.
Proteins, DNA, and RNA, and things like that, right? So molecular biology. And there are people who study the structures of these molecules. And this end of the discipline is very, very close to physics and chemistry. And it involves a huge amount of very sophisticated experiments to determine protein structures and so on. And in fact, the biggest breakthrough in AI in terms of impact on biology happens at this end of the spectrum.
Sure.
AlphaFold, which is now able to give reasonable guesses about what a protein structure will look like just from the sequence. Then there are people who study how these proteins act together in systems that work inside a cell.
Okay.
And so I work in sort of that kind of area. What goes on inside a cell? How do all these active processes work to allow a cell to replicate efficiently or to find food and to optimize its metabolism and so on? So these are like networks of proteins. And so people who work on that will be considered sort of cell biologists. And subsets of cell biology will be, stem cell biology or microbiology. It depends which species you're studying, right?
Sure.
And people who study signaling networks are sort of halfway between cell biology and molecular biology. There's that group. Then there's the level of tissues. And this applies to multicellular organisms like us, animals or plants. We actually start life as single cells, but then develop into this beautiful macroscopic structure. So how do individual cells grow, divide, coordinate, and shape themselves and specialize to create a whole organism? And that entire field is called developmental biology, the development of an organism. It is a very, very large field. This field begins to touch upon also issues of human development. What are the errors that can happen when organisms develop and so on?
Just to go back, the first two, molecular biology and cell biology, are more or less organism or species agnostic, or is there a species?
That's a very good question. So it's one of the huge discoveries of biology that at the very, very at the level of DNA and proteins and so on, biology is universal. Absolutely universal. I can take a piece of DNA from a human and I can put it in a bacterial cell, and with very few modifications, it will express and potentially do something.
Wow.
So it didn't have to be this way, but it turns out that all life uses a common language of the genetic code, the amino acid code, a certain way of making proteins. So at that level, biology is universal. At the level of cells, it is also one of the major discoveries in biology that the organization of cells, there are only 3 different major categories. They're called archaea, bacteria, and eukaryotes. We are examples of eukaryotes. There is a universality to studying biology at the level of a cell. If I study one eukaryotic cell, I can extrapolate what I do to most eukaryotic cells. Not all. There are always exceptions. But you can make general statements and then you can classify the exceptions. When you get to development, it is very, very species specific. But of course, there are commonalities. You've seen the famous pictures of the embryo of a chick and a rat and a human and even a fish. And at a certain stage in development, they will all look very, very similar. But before and after, they'll look very different. And beyond that, of course, it becomes very, very, very species specific.
Sure.
And this is one of the difficulties, right? You do experiments where you take a drug, which looks very promising in the lab, and you test it on rodents, and it looks, again, good. When you test it on humans, it doesn't work at all. So when it comes to, for example, applying biology discoveries to human health, the species differences become quite relevant. I'll just use this point to say that in the 20th century, there was starting from the beginning of the 20th century, there was the idea that the earlier approach to biology, which is studying anything you saw, just study the organisms around you, and whatever it was, geographically very different, and just capture insights from all that, yielded to a different way of doing biology, where people said, let's now focus our efforts into a few, what came to be called model organisms.
Sure.
Right. So you have the fruit fly, or the C. elegans, the worm, or you have zebrafish, or Arabidopsis, the plant. Right. A few. A very important step, because then a lot of different people could pool their efforts and work in different labs around the world and still make common contributions to understand how things work. Right. So that is how the huge advances.
But underlying that approach was this discovery of universality.
Presumably. So this was one of the ways they discovered it, right? But when you read these papers which say that in mammals stuff works like this, and if you actually read the paper, they mean they tested 2 rodents and 1 human stem and human cell line, and they say in mammals, right? Okay. What's changed in the 21st century because of things, first of all, like CRISPR, which allow us to knock in and modify genes in any organism, and secondly, because of huge taxonomic surveys that have been going on around the world. You just lift up deep sea samples or mud, or you go up to the Arctic and dig up frozen ice, and you sequence everything, right? So now we have an idea that the level of diversity on the planet is much bigger than, of course, we have ever sampled. And because all that diversity can be classified according to Darwin's, dictum of life being on a tree, we realize that some parts of the tree of life have not been sampled. So these days, a young scientist starting off can choose to put their flag in some totally new territory and say, I'm going to develop a new organism to study because it's so weird and different, and go and work on that. And this is not possible, 15 years ago, right? Wow. And so now the level of crazy biology that's coming in is at a much more rapid rate than it used to be.
Sure.
And so things we took to be universal now are less universal, because you find more and more outliers.
I see. Okay.
So this is very fun time.
Where I interrupted you. Development
biology. So development of tissues. And then, of course, if you can study what's called physiology, which is you have an organism, it's not developing, it's already there. How does it function? How does the circulatory system function? How do organs function and coordinate with each other? There's a lot of work on how different body systems that previously were thought to be independent, like the gut and the brain and so on, are really quite interdependent. And disorders do have diverse causes across the whole body. Very, very difficult area. A disease like diabetes or insulin resistance, these kinds of things have systemic causes. and it's hard to put your finger on one thing. And once you get to this part, you're slowly getting into also clinical practice. And I find this to be a field where we still don't have the basic phenomenology set and measured to the point where we are really satisfied with our level of predictive power and the ability to guess what will happen when you do some intervention. It's still very early days. So
Sure.
That's physiology. Sticking with that direction, I guess there's the brain, the study of the brain, which typically means the human brain. But that is almost its own independent field. I would say there's biology and then there's neuroscience.
Wow.
The study of how the brain works. How brain cells work is, of course, a cell biology question. In fact, a lot of cell biology was discovered by studying neurons, because neurons are such extreme cells that certain processes are much more evident in neurons, like the discovery of motor proteins happened in neurons because they have to transport things across this whole axon, right? So there is cell biology in the brain, but then there's the question of how the brain works, which is its own field. So there's that. The bit that I haven't talked about, which is sort of parallel to all this, is ecology, the study of the natural world, the history of organisms on Earth, the study of how collections of organisms work in ecosystems. It's a field biology. Intersects with things like climate change, intersects with human-induced change of other types on the planet, habitat loss, and so on. So that's ecology. And then there's the bit that holds it all together, which is evolution. It's
And it's still an active area of research.
No, of course it's an active area of research. I mean, so it's the broad strokes of Darwinian evolutionary theory still play out in very different specific ways in various cases, right? So evolution is still very, very central to how biology works, and any biologist will have more insight into their system and question if they take the trouble to study evolution and how it has implications for what they do. The sad thing is that it's taught as a parallel track, and mainly you leave it once you get to college. You know, maybe most of the evolution people have studied, even if they're biology students, would have been in school, and you never return to it again, as if it's a fully developed and done subject, which it is not at all. We still don't understand how the basic forces of evolution play out in specific cases.
Sure.
And how do mutations actually arise and create benefit for the organism? How do they spread through the population and sweep and get fixed? And how does this intersect with other organisms competing in the same environment? Very little is known.
So this is a wonderful taxonomy based on length scale of organisms, and presumably the complexity grows with their length scale in some sense.
Well, I wouldn't say that. I mean, I think you'll find complexity at every length scale. The complexity of how a protein folds intrinsically is a complex issue. The way to think about it is that the complexity of that problem doesn't cause difficulty at higher problems. That is, if you assume that proteins fold in some way without understanding it, you can still use that as the basis to study the next level and the next level. You don't have to have an atomic theory of something to understand how an organism develops, right?
Okay. This clearly lays out pathways for researchers to go forward. Let's talk about where the industry is and where some potential careers for life scientists in the industry. I started the conversation with vaccines. Did life scientists play, or do they play a role in something as simple as vaccine?
Of course they do. I mean, so vaccines are one of the triumphs of research in life sciences. They came out of the study of the immune system, which is, a mind-blowing, collection of things that our body is able to do using these cells of the immune system, the white blood cells of various types, the antibodies on their surface. That subject, you can find parts of that have molecular biology, like the antibodies, cell biology, systems level biology. So just the immune system straddles all these length scales. And the idea of vaccines came out of the phenomenology of vaccination was independently discovered.
Sure.
That people realized that you could sort of use infected samples to create a certain resistance to further infection. But figuring out how that worked and then using that to develop vaccines to new diseases is something which is a triumph of the life sciences. But your question is about careers. I should say that I'm speaking as an academic in the life sciences, and my main knowledge about this is sort of academic oriented careers. I do, of course, come in contact with industry during the course of my work. But what I say about industry, maybe an expert would have more to say on that subject. As an academic, of course, you can become a professor and teach and do research in the life sciences if you're at a teaching institution. Although typically your level of research will be lower the more teaching heavy the institution gets. At a place like IIT Madras, of course, you have to keep both those balls in the air as you juggle your teaching and research. And where I work at NCBS, which is part of TIFR, it's much more research heavy, and we typically teach only masters and PhD students. And in these fields, you typically specialize during your PhD and your postdoc, which of these kinds of biology you're doing, developmental biology or immunology or neurobiology or whatever it is, and eventually start doing the work. In industry, in the life sciences, there are two levels. One is the basic industry that provides the machinery and the reagents and the resources to just carry out fundamental research in biology.
Okay.
The people who make microscopes, people who make other kinds of analytic tools, the people who make fluorescent probes to study what's inside cells, the people who just make the basic reagents, the chemicals that we use on an everyday basis in a biology lab. So that entire cluster of requirements is an industry on its own.
Okay.
It's a platform industry. DNA sequencing, for example, is comes out of many companies these days who keep improving on the reducing error rates of DNA sequencing and reducing the cost per base of sequencing. All that happened in industry, not so much in academic labs once the technology was transferred out. So that whole level of industry is there, and there are lots of large companies that are involved in this, including the diagnostic equipment that's used in hospitals all the way down to the equipment that's used in labs. There are startups that are also involved in this that eventually maybe get acquired by large companies. There are many startups in Bangalore, for example, that are incubated at C-Camp, which is on the NCBS campus, which are involved in building new kinds of tools to study living systems. So that's one level. Then there's the pharmaceutical industry, which, I think enough, I'm sure your viewers will know enough about pharma because you read about it. Their goal is very specifically to develop drugs, which is to say, chemicals or protein-based drugs, which can be used to treat specific diseases. And over there, the broad philosophy is that there's some complex disease. After a lot of research, people find out that maybe this particular molecule is involved in this complex disease, and that would have been work that happened in some fundamental lab. But then somebody decides, maybe if I had a drug that prevented this molecule from doing the bad thing it's doing, maybe the disease will not have such a strong impact. And so you basically find little molecules that bind to bigger molecules, and then once you've found that, you make sure it doesn't have any toxic effects. And once you've done that, you make sure that it works in rodents. And once you've done that, so there's a whole pipeline for pharmaceutical development. It's very, very mature, very well established industry. I think, not being an expert in the internals of how that industry is, of course, they employ a huge number of life scientists. That is, people with degrees, undergraduate degrees, BTechs, PhDs, postdoctoral stints. So they are a major employer. But I think what's different now than what it used to be is these large companies are surrounded by a cluster of smaller organizations which provide these platform technologies. Some are sort of small contract research organizations that will do, for example, computer simulations for these large companies in order to optimize their chance of success, right? So there are smaller kinds of almost startup-sized companies also that will be doing these kinds of things.
Okay.
And so one could go work with a startup. Of course, the stability is lower. That startup may or may not survive for 5 years, but it's exciting to work with a startup. And I think there's a large enough ecosystem in certain cities Hyderabad or Bangalore, for example, where people do find a good launch pad for their industry careers coming out of a PhD in one of these smaller organizations. So there's that industry. There's an entire industry which I'm not very familiar with, dealing with plants, agriculture, and optimizing breeding and optimizing traits in plants. Since I'm not familiar with it, I won't say much more, but they exist obviously and serve a very important function. So there's a gamut of opportunities.
If you look at a parallel between software, computer science related professions, the massive explosion in careers in India happened because of multinational companies coming in and setting up what are now called global capability centers. Do you see that kind of a thing happening in life sciences in India?
It won't work like that, right? So I think it's a mistake to say that India had this software boom fueled by, there were multiple ingredients to making this happen.
Sure.
The timing was right. We did have an early advantage in terms of training, even from the 1980s, people to program, and so we had that the basic human resource capability available, and it scaled into a very huge thing. Software infiltrated every industry, and so it needed a back end for that. I don't know the numbers offhand, but the total scale of life sciences will not be anywhere near the total scale of manpower required for software, and the uptake will be much smaller. So I don't think one can take the government policies to promote software development and just copy and paste it into policies to promote careers in life sciences. Also, the level of investment for a company in, let's say, pharma or certain other kinds of development or diagnostics, the investment is much higher, the failure rate is much higher. So you don't have so many startups as you would say in a software industry, where it's easy to try and fail and you move it to the next thing. This idea of founders in software and the whole Silicon Valley myth, right, or mythos. I don't think such a thing can, at the same scale, can be sustained in the life sciences.
Even if it is at a smaller scale, I'm imagining a Pfizer or a Moderna or one of the large pharma companies, a GSK, coming in and saying, I'm going to do some of my wet lab work. Of course, I know for sure there's a lot of AI and data science work related to these companies that's already happening here. But is it possible for that something like that could happen?
I don't know. So this is beyond my pay grade. So I don't know. So of course this would have to be based on the economics of doing this. But again, I come back to the point that a person who is well trained in doing wet lab sciences, it's a lot of investment in their own education, both time and resources, before they get to the point where they're able to start off in a company like this. So if you're training people at that level of quality, then maybe the manpower will exist to do this kind of thing. If we do not do that, then a degree in biotechnology where you've only had access to a lab for a very short time of that time, you will not be prepared, and a company may not want to take the risk of taking you required, giving you training and so on.
Good point. So what I'm taking away from this chunk of conversation is that software industry had a more ripe human resource for the industry to come and harness value out of. In India, in life sciences today, that is sort of the rate limiting step.
I would say it's a major rate limiting step. Having people who are capable of coming into a lab. It's also a general issue, I find, that theory is considered higher than experiment in general, right? So if you're in school and you have the ambition to be a topper, then you tend to want to do more theoretical disciplines than getting your hands dirty or tinkering. And this lack of tinkering really shows once we get people at this stage. If you weren't a tinkerer, let me put it this way, if you were a tinkerer, you'll probably do quite well in learning how to do molecular biology. We need to encourage those kinds of people, and we tend not to.
Okay, good. I think we see that if there is a large, or when there is a large trained workforce in life sciences, this could be a potential opportunity that could open up for India. I think that
maybe it all depends on the business case and the economics of it also.
Sure. Let's switch gears and talk about AI, data science, and all of these, I'll call enablers that are around us as we speak. Um, the traditional model has been learn AI as a computer science student and somehow figure out how to apply it to a problem in a different domain. If you are, would there be a case to say, let's teach all life scientists some bit of data science and potentially some bit of AI?
So it's a tough question, but I'll speak by analogy. So one thing I do believe that all life scientists should know these days is, ability to code little bit on your own. If it is just writing a script in an Excel spreadsheet to do a little calculation of your own, that's also okay. But the idea that you can interface with the computer and make the computer work for you, rather than just consuming what the computer is doing. And most people who do PhDs now, they'll do their calculations on their own. They'll do statistics on their own. Most people who work with cells do the image analysis on their own. Okay, they don't, of course, write the image analysis software, but they write the scripts on that software to do the analysis, right? So it's become, just part and parcel of what you do as a biologist. If you look at the advances in computer science that made a big impact in biology, one example is a tool called BLAST, BLAST, which when the first DNA sequence databases and protein sequence databases were being put online, the idea was that people started sequencing genes and proteins, and so they needed to be put into centralized databases that could be searchable. But, I have a new gene that is implicated in my experiment, and I want to see, has anybody seen this before? So it's a string alignment problem. What a string means, it's a word, a long word of letters. That's a computer science meaning of the word string. So I want to see if my gene has the same letters in the same order as something else, but it's not exact. And so this tool called BLAST, which was developed by computer scientists, was a very rapid string alignment problem that could be run on an entire database with your string on an online server and give you the answer in seconds. And once this came in, every biologist, when they came across a gene, they would take the sequence, they would do BLAST, right? It became part of the protocol. And it was it was a game changer, because then you could do gene discovery much more quickly. I know that my gene is like your gene, and you've already published papers on your gene meaning the gene you study. And so I accelerate my research. As you say, it's an accelerator. Similarly, phylogenetics, the ability to see the relationship between organisms using quantitative traits, optimized by computer science. I already mentioned AlphaFold, which is how protein structures are. So biologists, by and large, if a tool is available which is computational, which provides important insights for their work, they will be the first people to adopt it. They always do. And so they'll even help you improve the tool or optimize it or suit it for that purpose if it is not already suitable. So they won't, of course, by and large, make the tools, but those tools become part of their approach, always.
So having a grounding in the math associated with these tools.
Okay, good question. So do I need to know how BLAST works in order to use it, right?
Yes.
You can be a power user of BLAST, in which case you'll have slightly better success rate than if you're an average user. But the average user already has huge success rate, right? So I would say that even with AI, we're still in early days. In a matter of 5 years, AI will be usable by non-experts without needing to know what's under the hood, by and large, except for edge cases where you really need to understand exactly what the thing is doing because there are some errors that will creep in otherwise, right? I mean, I certainly don't, when I write a simulation and I have to have an ODE solver or something, I don't care which ODE solver I use. I just pull it off the shelf and I run it, right? Or PD. So I trust that these solvers are supposed to do what they say on the box, and I use them. And it would be not worth my time to go in and try and recode that whole thing. And similarly, I don't think one needs to have an understanding of such sophisticated tools. What you do need an understanding of is a basic understanding of numbers and the fact that numbers matter, right? And that part, I think we don't do a good job of. So there's a very nice book by Rob Phillips and Yannick Kondev called Biology by the Numbers. And the ambition of this book, when you learn physics as an undergrad, one of the first things you buy is this table of constants. And you know there's only a few constants, right? But from those few constants, you can figure out a huge number of things, including the size of a star, and including the size of a gene. You could even figure such things out. So what Rob Phillips is trying to do is to say that every biologist should have a facility in taking numbers, which are constants of nature or measured empirical things, and mixing them up in their heads and coming up with hypotheses which are not qualitative, but they're also quantitative. That is much more important than knowing how a GPT transformer works. I don't think that's important at all.
Okay. Is there a case to be made, given how pervasive AI is likely to be a few years from now, that every BSc student out there at least learn from an early age how to use these tools?
Yeah, well,
it could even be as simple as taking a little bit of a formal course in not deep learning formally, but at least the use of AI within their domains.
So I would find it surprising if students didn't spontaneously go and learn about this stuff, right?
Yeah.
So it seems that this will happen very naturally. And one thing I missed, by the way, earlier, the question of if you want to get into life sciences, whether you have to study life sciences, the answer is no. You could come from AI.
You're a case in point.
Yeah, I'm a case in point. But you could come from AI, and you could get. So life sciences is a field which is also moving so rapidly that even if I did study life sciences in college 10 years ago, the set of things I would need to know now is a whole other college degree, right? So there's no downside to coming to this field late.
I see. I'm going to ask a question on that, follow up, as a layperson. When I open a random life sciences paper in one of our archival journals, the starting point is very daunting. The starting point is very...
Sure. It has a lot of jargon.
It instills a sense of fear.
Yeah.
Help me there, please.
Yeah, but have you tried reading a math paper? So,
so it's worse.
Consider the p-adics, and then poof, yeah. There is a reason why formalism is important and rigor is important, and these papers speak to experts. Yeah. The way I deal with this, and this is the way every life scientist also has the same problem you do in a field that is not exactly their own.
Oh, within the life sciences.
Right. So it'll, you come to every paper assuming that, you're not going to get through even the abstract, let alone, pass the introduction, right? Going to a talk is a little bit better. Maybe you'll get through the first 10 minutes. So this feeling you have happens to everybody. The field itself has developed an entire system by which knowledge is transmitted from person to person. And, for example, these review articles are very important in life sciences.
Yeah.
So you ask an expert to look back on their field for the last 5 years or so and to summarize it in a language that other life scientists can understand. Forget non-life scientists, right? And so you read the review, use that as your entry point, right? I've changed fields in biology, at least I got into it studying bacterial gene regulatory networks. I had to read it from the textbooks and then read papers. Then I started looking at eukaryotic cell biology. I had to learn a whole set of things. Then evolution also. Each one of these times, I had to pay buy my ticket by doing all these things, right? The language is daunting because it's written for very specific domain experts, but there are resources that are available to help you deal with that.
Okay, good. A little more sort of a nuanced but fundamental question in students' minds. What is the difference between a BSc in biology, biotechnology, and now there's bioinformatics?
Sure.
And how would I know which one is the one I need to choose?
Sure. These terms, by the way, work very differently in different countries. So in Europe, for example, the term bioinformatics it's much broader. It covers all aspects of systems biology, quantitative modeling, and sort of computational bioinformatics like BLAST. I would add to that list basic degrees in a number of disciplines. So first of all, even in biology, typically, you would have specializations in microbiology,
In
botany,
zoology.
zoology,
Yes.
biochemistry, and so
Thank
on
you.
and so on and so on.
Yes.
Okay. I find that students who have been trained in some of these more classical disciplines as undergraduates have a much more solid understanding of that part of biology than people who have more generalized degrees.
Okay.
Okay, so if you had to start off by studying life sciences, I would weigh in the option of studying one of the more classical disciplines. The reason is that because the pedagogy in those disciplines is very mature. They sort of teach you how to think about biology. It doesn't matter that the set of phenomena that they're talking about may be a little bit out of date, but they teach you how to think about biology.
Okay.
Biology is all about thinking. It's about you have a puzzle and you're trying to figure it out, and you have totally incomplete information. So when you study these fields, biochemistry, you have a historical account of it. How did people figure out what an enzyme was? You learn all that. And that gives you a roadmap for how to do new discoveries in biology. So I don't think even in a general biology degree, you would take courses of these types. Biotechnology is a bit of a funny beast. I think it came out of the desire to replicate the software boom in the field of life sciences, right? And to generate a huge amount of manpower that would be trainable and would be able to work in companies. I don't think it's lived up to its promise because, as I said, you don't have the resources to train so many people with the experiments that they would have to do to become proper bench scientists. And the other problem with biotechnology is that you do of course learn basic biology, but not enough of it. Very quickly you learn about the applications of biology. You learn about PCR and CRISPR and all. But those things are very fragile because they go out of date. I mean, if you're trained on it's like a car mechanic who's only specialized in Fiats of 1970/1980. Once the new car comes in, you don't have anything to fall back on.
Yes.
So your foundations were not strong enough, right? So the curriculums in biotechnology in the good places avoid that problem. But in a place where it's more heavily tilted towards making people employable, that knowledge tends to be more fragile and goes out of date quickly. To people who are in such degrees, I would say it's very important that you spend a lot of your time also in parallel using online resources, of which there are a huge number,
Sure.
to really solidify your foundations in biology. You may be learning how a certain machine works, a chemostat or whatever, but also try and query, how people invented that, what are the principles, and build up your own knowledge while you're learning this.
Sure.
It's actually quite easy to pick up all this on this side. So that's biotechnology. Bioinformatics is a specialized discipline, which I think you learn a little bit of coding, you learn a little bit of math, you learn a little bit of everything. My summary for all these things is I believe these more specialized fields should always be master's degrees.
Okay.
And that the undergraduate degree should be more foundational and conceptual.
And so that brings me to the next follow-up. It almost seems in life sciences like an undergrad degree by itself is today not a currency that's accepted in many places.
I tend to agree with that for all the reasons we talked about already, because unless you've had exposure to the experiments and been a tinkerer or had a real good grounding in the foundational aspects of it, on its own, the undergraduate degree in life sciences may not be beneficial. I would just do my undergraduate degree in anything that I find exciting and I'm passionate about, even if it's, let's say, mathematics, even if it's economics, right? And one can get back into biology. Concepts from economics can be applied to the understanding of ecosystems, for example. Sure. So, don't over-plan it and say, I want to get into this and therefore I'm going to do a degree in this. Keep your options as broad as possible for as long as possible while still doing a subject you find interesting. That's much more. And I would even say, if in your college there is a teacher who's exceptionally good in a field which you didn't think you would be interested in, I would choose that one rather than, some subject just because it's a life sciences subject.
Sure.
Yeah. And then by the time you get your master's degree, you'll have a much broader understanding of what life sciences is. You'll make better choices, but whether you want to study immunology or neurology, neurobiology, you do that in your master's and then in your PhD. There's a reason why that master's is a valuable transition point.
And then what does a PhD add to this? Why would somebody do a PhD and when?
So I my brother wanted to do a PhD in math. He loved math. He studied math as an undergrad, and I hope he doesn't mind that I'm telling this story. I had a book about the title was How to Do a PhD, which I found at one of these book fairs in Bangalore. And chapter 1 of the book is Why Not to Do a PhD, right? And in the end, he decided not to do a PhD. The PhD is it serves only one purpose. It is to train you to become essentially an academic researcher, potentially an industry researcher, but of a certain type, right? It is not for everybody. It's not needed. It is extremely taxing on the people who go through it. And if you don't have the emotional resources and the passion and everything to get through all the ups and downs and the immense duration of the PhD with very little payoff on the other side, what it takes out of you, I don't think is worth the payoff in general, right? That said, these days, though it wasn't true 100 years ago, the PhD is a requirement if you want to become an academic, if you want to become a lecturer, if you want to become a researcher at a university or educational institution. You have to do it. So that's one reason to do a PhD. If you're sure that that's the career path you want to go on. During my PhD, I many times wanted to quit because things were very difficult. They were not working. You see other people who have success, and you're not quite there yet. Your system it's overwhelmed you. You don't understand what's going on. So it's something which I do not recommend for everybody. I mean, you have to have a good reason why you want to do a PhD. I think it's true that in industry, those who get hired after masters versus those who get hired after long PhD plus long postdoc, eventually the payoff, even in terms of total salary accumulated over the years and so on, doesn't seem to be commensurate with the amount of time it takes to do a PhD. There are articles published about this every so often. But if you're the kind of person who, just cannot go to sleep because this problem is obsessing you, right? Well, then you should do a PhD.
Yes.
Yeah, but you should know you're that kind of person, and you should know how difficult it is going to be.
Right.
And hopefully the institution you're studying in gives you all the resources you need to get through this huge labor and come out the other side, happy. For me, finishing my PhD, of course, looking back, I learned a huge amount. I learned how to ask a question, how to design an experiment. I met very interesting people with whom you could talk to at a very high level, all that good stuff, right? And I love being a scientist now, which I would not have been able to do if I had not finished my PhD. So it has a function, but you should know that that's what you're setting yourself up for. You should have a very clear idea of who you yourself are and that you want to do this. And with your eyes fully open in a way, you should go and do it. And not be afraid to quit at any point. If you don't like it, don't stick around.
And you'd still have come out ahead
sure.
not even starting.
Sure. The idea that you have to finish and so on is totally meaningless.
Oh, that's a wonderful insight, actually. Starting a PhD is always a good idea. Finishing is optional, meaning make it optional in your own minds.
Sure.
Especially because, in life sciences, you had said earlier, there's no recipe, do 1, 2, 3 and, write down your result at the end of it and and you're done. So given that, you have to give serendipity a chance. And
fair enough. So
failure
maybe try it out.
And rejections are part of life in the life sciences, so to say.
Well, very good, but you shouldn't start off by saying, I'm just trying it out. You have to really believe that you're going to get through it.
Okay.
If you just start off by saying, I'm trying it out.
Is there a systemic a way to learn this resilience to accept failures, or how it seems like life sciences requires more of it than even physics, say?
Yeah. I again raised the example of mathematics, which is much, much harder and saps you, dry as you do a PhD more than in physics. There's a new book by Chandrasekhar Khare called Chasing a Conjecture, which I just finished reading, where he describes his journey of failure after failure after failure during his PhD and his early faculty career before finally stumbling onto something. And in retrospect, one could ask, was it all worth it? And I think some people would say yes, and some people would say no. But learning resilience, it's a life skill, and uh, we all pick it up for different reasons and different ways. And you don't have to put yourself through the struggle of a PhD to learn that. But it is a very important thing that I think people who have that end up being successful. How they picked it up, it may have been intrinsic, they may have learnt it. I know I've never thought about it as something that can be very easily taught. I think you have to go through many things and develop it yourself.
Okay, let's look into the crystal ball. Let's gaze into the crystal ball and see what are some new ideas that could become careers a few years from now, maybe a decade from now.
Yeah.
Where is the world of life sciences going, both in research and industry, as best as we can?
Yeah. One thing we haven't talked about is this intersection of the life sciences with material science.
Great.
So there's a lot of interesting stuff happening now in what used to be just chemistry departments doing chemistry of liquids, and now they're making programmable materials, right? They're designing matter from scratch with very, very sophisticated properties which require an understanding of the system from the atomic level upwards, right? I think that field will have a very productive collision with biology, if it hasn't already done so, very, very soon. There are already people thinking about how cells interact with their substrates and the mechanical properties of those substrates, and how they can be used to differentiate cells into specialized states. And so there is I think, and for example, 3D printing very, very small patterns and having tissues grow in those patterns, these kinds of things, very exciting work has already happened and much more to come. And that way you bring in the entire knowledge of chemistry, which is so mature and so immense, and bring it back into biology, not thinking about what's happening inside a cell, but building environments for cells to do interesting things in. So I think it'd be huge. Of course, I won't talk about AI. As I said, I think AI we'll just become users of AI. We're not going to develop it, and we won't need to. Just like we're users of computers without knowing how computers work. I think that the planetary questions, of course, are going to become huge. That is to say, global change, ecosystem change, how it impacts things at every level. I mean, what people don't realize is the change of 1.5 degrees on an average actually moves the tail of the distribution enough that events that used to be exponentially rare now happen, once every 10 years or something like that, right? And I'm not talking about extreme storms or things like that. Just, in a given ecosystem, some insect used to not be able to grow there because the winter temperatures used to be somewhere, but now that insect is growing there. These changes, I think, even to document them, because they're happening so fast, is an effort that requires huge amount of global coordination and people coming in from different disciplines, from economics, how humans interact with these things. So biology, through the lens of ecology, impacts how humans are able to carry on their livelihoods, how resources are going to be distributed in a fair and equitable fashion. So people who are trained in even without any training in life sciences, but who are, for example, trained in economics or social sciences or these kinds of disciplines, I think will have a role to play in taking insights from biology and applying that to how humans can live more peacefully on this planet. That will be something I would suggest any student start to think about.
Okay.
Right? Global issues of this type. And I can think of a dozen more things, but you asked for some two big things, so No,
I think this is there is enough happening at the interface of environment and life and matter and life, that I think those interfaces now seem to be exciting spaces for new research, new ideas, and possibly.
And if I can just really speculate, I think before the end of the 21st century, we would have discovered strong evidence for life on other planets, right? And so that's something which I find it'll be the biggest discovery humans have ever made. And that's something which I think,
Okay.
a student who's passionate about could think about.
Let's come back to research. I want to talk to you about a documented problem in life sciences, which is replication,
Sure. Sure.
failure to replicate results. Sure. What is the stem of that problem?
I mean, it's a question that requires a nuanced answer. There is the so-called replication crisis. That is, you do an experiment and then another lab reads your paper, does the same experiment, and they don't get the same result. And in 90% of the cases, it's got nothing to do with 99.9% of the cases had nothing to do with fraud or misconduct or anything like that.
Sure.
Okay? So we won't talk about the fraud issue, which is
Sure.
there are some bad apples, but it's not the main issue. The main issue is that there are incentives in academia which are totally counterproductive to publish papers. My promotion is determined by papers I publish and which journals I publish them in. And so these crazy incentives force people to, publish more flashy-looking results than less flashy-looking ones, or publish in journals that have less rigorous peer review than otherwise, and so on. Every epoch of science, it has always been that people try and do experiments, and in retrospect, their conclusions were wrong, right? And so the process of weeding out the incorrect ones and the correct ones is a healthy aspect of doing science. When you could just ask, though, do there tend to be more of those incorrect conclusions in the published literature than there used to be? And the answer is yes. And the reason is just these incentive structures. If we just went back and said we don't care about the quantity of papers you publish, we care in some sense about quality, which is hard to judge, and had a better way to judge progress of an individual, their institution, a national level scientific enterprise, better ways to judge how well these things are doing than just counting papers, this problem would more or less go away.
Is there a more fundamental issue with variability in biology that we today don't understand?
I don't think so. I think if you say you did an experiment, so for example, in the 1940s and 50s, these amazing experiments in microbiology, looking at viruses and bacteria, how they interact. Once people narrowed down and said, I'm going to study this organism, this strain with these conditions, then experiments done in different labs at different times all across the world, you could overlay them on a single graph, right? Life sciences experiments, but how often a bacterium mutates, and you get a perfect straight line. Okay, so it doesn't have anything to do fundamentally with something crazily uncertain about biology. I mean, I don't think that's the case at all.
Okay,
you can do a biology experiment that should be replicable.
Okay, that's a good lesson to leave for all of our researchers to chew and digest. I think there is. This can become to harm the whole enterprise, the science enterprise itself, if we are not careful.
Yeah. So, the way I look at it is that the enterprise of science is going on, and the honest errors are the tail of that enterprise, and that's perfectly fine. It's a healthy aspect of doing science. Then you have really shoddy work, and then you have fraud, which are the minority. And I think the shoddy work and the fraud, there are things called paper mills where people, they publish fake papers in fake journals with fake editors, and they get points on their CV for doing this, right? It's very clear to me as a reader that this paper is not worth reading, right? So I don't think it is so easy to contaminate, even though these things exist, right? The progress of science. So these are two parallel tracks, right? Shoddy and fraudulent work and genuine work with honest mistakes on the side and wrong speculations or whatever. I don't think this will overwhelm, even if the number is large. Anybody who's actually a practicing scientist, who's been doing something long enough, will know where the boundary is.
Okay. Okay. Good. So let's wrap this up. What's the one piece of advice you want to leave with every young student out there, sort of interested in a career in life sciences? How would they parse their own thoughts?
Yeah. So I come back to keep your options open for as long as possible. Do a degree now that allows you to think about what you want to do later, because you don't know who you are now and what you may be interested in a few years and where the world has been going in a few years. And so these very basic classical degrees, I think, are very good starting points for you. There are ways to discover whether you like life sciences these days. You look online, there are so many resources, right? So you can discover for yourself whether you're the kind of person who would enjoy doing these kinds of things. The number one thing I would say is, please be a tinkerer. Try and get your hands dirty. Go and do an experiment, even if it's just outside in your, you pick up some mud and put it under a microscope. And these days you can get microscopes in your own home. It's very, very easy. Learn skills of observation and do that separate from your degree. It's a skill and I'm sure that kind of thing will keep you going for a while.
Fantastic. I hope this excites more young students to take to, a more breadth of careers in India and in including life sciences. Thank you very much, Mukund,
Thank you for the opportunity.
This wonderful conversation.
It's been a great discussion.
Things, and I hope it remains in the archive for students and parents to use.
Thanks.
This has been another edition of the Professor Mahesh Podcast, your resource to help you make informed career decisions. Thank you again for listening. Until we meet again, be well, do good, and keep in touch. Goodbye.